Empathy. The ability to share another individual's emotional state has been suggested to be a source of prosocial motivation by attributing negative value to actions that harm others. In other words, harming other people is unpleasant because we vicariously share the pain we inflict. Humans are not the only species that display effective reactions to the distress of conspecifics. Recent studies suggest that rats are sensitive to other conspecifics' emotions, a phenomenon that could rely on recently identified emotional mirror neurons in the rat's anterior cingulate cortex, which respond to both experiencing and observing pain. We developed a paradigm to assess whether rats avoid options associated with others' pain. In this paradigm, a rat called the Attah can press 2 levers for the same amount of sucrose. After a baseline phase revealing the rat's preference for one lever, we pair this preferred lever with a shock to another rat, the victim, located in an adjacent compartment. We then measure how much actors switch away from the preferred lever as a behavioral index of harm aversion. We find that rats reduce their usage of levers associated with harm to a conspecific. This effect is stronger when actors have been exposed to pain before the experiment, and is observed in female and male rats, as well as for unfamiliar victims. Furthermore, we show that rats reduce the usage of the harm-inducing lever when it delivers twice, but not thrice the number of pellets than a no-harm lever. Finally, we show that pharmacological deactivation of the anterior cingulate cortex, region essential for emotional contagion, reduces harm aversion. Taken together, this model of harm aversion might help shed light onto the neural basis of psychiatric disorders characterized by reduced harm aversion and provide an assay for the development of pharmacological treatments of such disorders.